pre-messenger RNA, or pre-mRNA, is a single-stranded RNA photocopy of an entire gene made from DNA, which includes coding exons and non-coding introns. Intron boundaries are marked by splice sites, with a GT at the beginning and an AG at the end. To make the mature mRNA blueprint for the encoded gene product, pre-mRNA undergoes an editing process called splicing, which cuts the introns out and pastes the exons together. Exons must be pasted together exactly right to keep the genetic code readable. Alternative splicing enables different proteins to be made from a single gene. Each intron has 4 specific motifs that are markers for splicing: the donor splice site that marks the beginning of the intron, and a cluster of 3 motifs at the end of the intron: the branch site, the polypyrimidine tract, a region enriched in CNT pyrimidine nucleotides, and the acceptor splice site. The splicing machine is called the spliceosome, a huge RNA protein complex which recognizes the splicing motifs of each intron to cut them out and join the exons together. The core spliceosomal factors are U1, U2, U2AF, U4, U5, and U6, which form shape-shifting supercomplexes together with hundreds of other proteins and small RNAs to perform the 2 splicing reactions. The binding of U1 to the donor and U2AF and U2 to the branch site and polypyrimidine tract is the step that commits an intron to being removed. U1 is the marker for the donor, acting as a guidance system that tells the rest of the spliceosome where to assemble. At the other end of the intron, U2AF finds the branch site and polypyrimidine tract. Then, U2 takes over at the branch site and scans to the right to find the acceptor AG. The tri-SNRMP is a complex of small nuclear ribonuclear proteins, or tri-SNRPs for short, which join U1 at the donor. U6 is the enzymatic engine of the spliceosome, and U4 and U5 keep U6 in an inactive state. U1 and U4 are displaced before the splicing reactions begin. U2 carrying the branch site swings over to connect with U6 that's with U5 at the donor. This movement brings the donor and branch site together in 3-dimensional space, with the exons on either side and the rest of the intron folded up underneath and out of the way. U5 holds on to the flanking exons and works with U2 to keep U6 under control until everything is aligned and ready for splicing. In the branching reaction, U6 cuts at the donor G, which is covalently joined to the branch site A, making the intron lariat. During exon ligation, the spliceosome keeps a hold of both exons, repositions itself, cuts at the acceptor, joins the exons together, and releases the intron lariat. Then everything disassembles and is recycled for another round of splicing.